Hi, everyone. Welcome to Navigating. Here at Navigating, we explore how people from all walks of life are finding their way through today's complex and changing world. One of the biggest challenges all of us have faced these past few years is the COVID-19 pandemic. Despite lockdowns and vaccines, millions of people worldwide have contracted the disease and millions have died from it. My guest for this episode has devoted his career to developing broad-spectrum vaccines. Jacob Glanville is the CEO of Centivax, whose mission is to accelerate the world's transition to a post-pathogen humanity. Hi, Jake. It's so great to have you here. Thank you.
It's great to be talking with you again.
I know you're a busy person doing the work that you do, and I appreciate you coming to talk with us about, we're kind of in a crazy time, you know?
We sure are, yeah.
There's politics aside, there's the things that are happening with public health. And, now we've got the bird flu that people are concerned about. And I forget, it's H what? You always talk like that.
Yeah, it's a H5N1.
H5, okay. And how, I guess, I think I'm going to start at the very beginning with you and how you grew up in Guatemala, right? Because your parents decided to leave the country long before the rest of us thought maybe it was a good idea. And you grew up in Guatemala. They ran a hotel and restaurant and.
Yeah, that's right. It was my grandmother's idea originally. She was a nurse, and she had 4 children that she raised alone. And so when she was old enough, my father was the youngest left. She decided to travel the earth, and she found this beautiful lake called Lake Atitlán in Guatemala. And over the next few years, she decided to build a hotel and a restaurant down there. And originally, my father came down to help her build it. And in the process, he met my mother. It was an artist from Los Angeles.
And she was living there already.
Yeah, she actually was my grandmother's roommate. So my grandmother connected the two.
All these nice tie-ins.
Yeah. And the way my father puts it is that she was trying to get my father to come down and help her with the complex process of building a hotel and a restaurant, getting land and hiring people. And so he told her, I'm too busy and my business is going on here. And she's like, oh, come on down. Besides, I have this, young blonde roommate you might want to meet. And he's like, oh, she knows me so well. And so he went down and turned out to be my mother.
That's all it took. Yeah. She didn't even send a picture, right? Yeah. Just the description.
Yeah.
When did you decide you were interested in medicine?
Yeah. So I think I was always interested in medicine. I had a visceral sort of Pavlovian interest that I was a pretty severe asthmatic. Mm-hmm. And so I struggled with it significantly because we were out in a Mayan village that didn't have a hospital within 4 hours, and the power would go out, which means I didn't have access to like a nebulizer.
Yeah.
And during rainy season, there's molds and things that make it worse. So I kind of always grew up thinking about that, where kind of, you take a medicine that makes it so you can't sleep. That's one of a medicine called Theodore. So you're a child, awake at night, not be able to sleep. And you spend a lot of time
because it hypes your system up.
Yeah. It's actually a lot of stimulants are good for asthma because they open the airways.
They open everything up. Yeah.
So yeah, you have this I'm hyped up kid. I'm super sick, can't sleep. And I'm up at night wondering why am I like this? Why are other people different? And so that was part of it. The other part was that the village had very poor medical access. And so we had a lot of giardia and worms and amoebas and all manner of tropical diseases that were reasonably common, which my whole family got. And I was always aware of that and interested in the immunology and the infectious disease aspects. And then I think 2 things that got me more interested were when I was 15, my father suddenly got very sick. And originally they took them a while to figure out what was wrong with him. They were thinking he had malaria. Then they were testing him for HIV. And then they finally found out he had this severe and aggressive autoimmune disorder called necrotizing vasculitis. And the effects of it are he lost maybe 80 pounds. He started getting gangrene in his fingers and toes and lost sensation and numbness in his feet and his hands. And we thought he was going to die. I was 15. I did what I could I was helping run the restaurant and the hotel while my mother was in the city taking care of him. And the remarkable thing about it was that he was eventually treated with the kinds of medicine you normally treat cancer, chemotherapy. And in most autoimmune diseases this doesn't work. But for this type, you can give them chemotherapy and it kills enough of the immune cells that it basically reboots the immune system to stop the
that's so
autoimmune attack.
interesting.
And so I think because, you know, obviously I was 15, so it was somewhat traumatic to be like, okay, I guess I have to take over now. And my dad, he was telling me he wanted to be cremated and things. So
Oh my!
that stuff stays with you.
It was to that point, yeah.
But there was also an intense, you know, just fascination with the immune system. And so I think that experience.
Yeah.
And then the last thing that happened was I went to the States for my last year of high school. And I was thinking about these things. By the time I applied to UC Berkeley, I had already written down I was interested in immunogenetics. And that's what I was interested in. And I was interested in computer science at the time as a hobby. I didn't realize the two were going to collide. And then the final stunning thing that happened for me was the first time I went back, which was a year and a half later, to the lake. At that point, it was, I think it was 1999. And so it had been a few years since the Civil War had ended. There was also better trade happening. And when I got back to the lake, I remember my brother and I were walking through the village, and we looked over. There's a basketball court in near the center square, and we saw these kids playing basketball, and we're like, "Oh, who are they?" And then suddenly we were both shocked, and we're like, "Wait, those are Zutuiles." And the reason we were shocked is that these people were our height. And growing up in the village, my entire time growing up, by the time I was 12 years old, I could stare across the entire market and see my brother. And because we were just a lot taller, the Zutuiles were much shorter. And I just, like, through ignorance, I assumed was genetics. That's just who they were.
Right.
And
Your
then,
dad's tall, whatever. Yeah.
But then the stunning thing, we looked around and we realized all the younger kids were getting taller than their parents in the village. And we realized that, and I'd asked the doctors what was going on, and they said, well, it's because we've introduced deworming medicine, and we have these like simple medicines were suddenly available in the village that they'd handed out in the schools. And it wasn't just that they were taller, like a head taller. The younger Zutuiles are much
healthier looking.
Healthier. They could pay more attention in
Robust
school. They were doing better. And that had, I think, an immensely profound impact on me, because it made me wonder, like, what else am I taking for granted now that if I look to a better future, we'd look back on and say, that was astonishing. It was holding us back. And so I think that kind of defined my whole career, where I you know, we like to think we're every generation and every moment we think we're living in modernity. But the truth is we are living in this ridiculous period where we put up with a huge amount of infectious disease that we just say, oh, well, that's life. You know, people are going to get staph and they're going to die. Older people are going to get coronavirus or pneumonia and influenza, RSV, HMPV. And that's sad, but it's what happens. And children will get all these diseases, and it's super avoidable. So I went to UC Berkeley. I got really interested in genetics, genomics, development, and immunology. And then along the way, I worked with two different laboratories, the Glynis Thompson HLA Population Genetics Laboratory that looked at these immune genes. They're the genes that decide whether or not you reject a graft, like if you're going to get an organ donation.
Okay.
Those same genes are the audit system that let your body know if any cell in your body is producing something weird. So a viral infection, a bacteria, a cancer cell that's making strange things. And we were looking at the genetic diversity of that all across the world for immune reasons, but also just tracking populations. And that was really fascinating, because that group was using computers and statistics. And this thing that was a hobby of mine, which was using Linux and writing programming, I suddenly realized I had a huge application in immunology.
Right.
And I worked with another group called the Berkeley Phylogenomics Group by Kemen Shalander. And she was really remarkable at developing algorithms to analyze populations of rapidly mutating proteins, which are basically is our immune system and the pathogens that fight us. We, as a species that reproduces like at best every 15 years, more often more like every 25 years, we have a challenge, right? Because we're confronted by pathogens that can reproduce every 30 minutes. And so they can mutate faster than we can.
Sounds like a losing battle.
There's vaccines, and then there's also just really understanding the power of the immune system. So the way we've survived these enemies that can change faster than we can is that we have beating through our veins and our bone marrow and other parts of our bodies, we have this population of T cells and B cells, these immune cells. And what they do is they shuffle up pieces of DNA to make hundreds of millions of new receptors, so proteins that can bind other proteins. And that's how we shuffle them up on a daily basis. And so that's how we, as an organism that only makes new children every 15 to 25 years, is able to fight off things that divide every 30 minutes, because we have this shuffling system to make novel, random rearrangements And that's how we make antibodies.
And
That's
we can keep, so let's say the pathogen is shifting and reproducing and changing every 30 minutes. As our immune system is shuffling away in there and something slightly new comes in, it's able to fight it off.
Sometimes, but sometimes the mutations escape.
Right, exactly. And that's what we've been dealing with.
That's the fundamental challenge. Our immune systems do a pretty good job of fighting off a single infection.
Right.
It has to do with how fast the thing mutates. If it doesn't mutate that fast, like chickenpox, you make immunity to it the first time. In that huge population of shuffled random antibodies, you're going to have some that bind chickenpox. Mm-hmm. And then your B cells have a way of tinkering to make them stronger. And then you store them in immune memory, which means the next time you're exposed to chickenpox, you already have B cells that go, I know exactly how to deal with this puppy, and you start spitting out these antibodies. And that can cover your lifetime. The reason people get shingles is really just eventually those cells die off.
Yeah, so I was wondering about that, shingles, because I know people who've had shingles shots and still get shingles. And that means that even the shingles shot didn't help the immunity enough.
That's because no vaccine is perfect. The challenge with that is we have some vaccines that are pretty close, I love vaccines. I work on them, but I think, and this is a kind of a bigger point, I think we need to be a little bit more forthright. And I've made an effort, and I've gotten harassed for it sometimes, but I've made an effort to try to be more stick to the facts about how the value of vaccines, but also their limitations. And the truth is, when any vaccine you make, you're injecting it and you're hoping that immune system will detect the useful parts, recognize the important parts, that then can arm that person to respond to the pathogen. But that doesn't always work.
Right.
And the other challenge is that as people age, they have this thing called immunosenescence. And immunosenescence just means that your immune cells are getting older. And so you need to give them extra kick in the butt. It's a little harder for them to produce a good immune response than a younger person's immune system.
Right. And so that's why older people are encouraged to get flu shots and things like that.
Yeah. The two reasons are their immunity wanes. And the second is that flu and coronavirus and RSV and HMPV, they're not good for anybody. And I think that what we're going to be learning, and we've been learning in the last 10 years, is that there are a bunch of additional side effects that happen that are causing long-term health problems for many of these infections. But the elderly are uniquely susceptible because their immune systems don't work as well. And second, that they are susceptible to getting a lung infection and then getting pneumonia. And pneumonia is one of the leading causes of death for the elderly.
Right. There's somebody right now at UC Davis Medical Center that I know because he started with the flu. It turned into pneumonia. And now he's on a ventilator, as far as I know. So getting back to your history, though, because there is a lot to talk about regarding what we're facing now over the last 10 years and what we're seeing. So when did you work for Pfizer for a while too, right?
That's right. So after UC Berkeley.
Is that something to admit or not? Pfizer,
I'm happy.
I know. Pfizer is always under attack somewhere.
So after UC Berkeley, I continued working in the Berkeley Phylogenomics group for about 9 months or 10 months after graduation. And then I got a job at this Pfizer site in South San Francisco. And at the time I got hired in at this time where Pfizer was changing from a pure small molecule chemical company. So those are the kinds of drugs like aspirin and Viagra and Lipitor and things like that. And they were getting an interest in this new class of drug called monoclonal antibody therapeutics. So we all know them from the pandemic. Those were those antibody therapeutics that Trump took during the presidency and were available from Regeneron and other companies. The Pfizer was realizing that they were a powerful new class of drugs. So you can make antibodies like our body does, but you purify a really good one and then you sell it as a drug. And it could be targeting a cancer. There's a bunch of pretty breakthrough cancer therapies. It could be used for autoimmunity, a number of other applications. Mm-hmm. And so Pfizer had created a site, or they bought a site in South San Francisco to advance the discovery of these kinds of drugs. And so I arrived, I had this computational immunology and molecular biology background in population genetics, but really I think they hired me because I could program, because they had about maybe 75 people when I arrived, and they had two ways of discovering antibodies, and both had some problems. Mm-hmm, one was they would get the drug target, so it's some receptor on the surface of cancer you want to target, and you think an antibody against it would help clear out the cancer or whatever. And then they would immunize that into mice, and then they would use this technology called hybridomo, which just means you isolate out a B cell that the mouse makes that makes an antibody against that. And then you have to do some engineering to take a mouse antibody and make it look more human, because otherwise you get an allergic reaction, or you get negative reactions to putting a mouse antibody in a patient.
So the B cells, that they tell me one more time about the function of the B cell.
Sure, the B cell is what makes an antibody.
It
Each
makes
B
the
cell
antibody.
makes a different antibody.
And then in the lab, then are they synthesizing an antibody that matches what they're seeing coming from the mouse?
That's right. Yeah, you isolate, because the mouse makes lots of different B cells with lots of antibodies, and so then you spend some time hunting through them to find the really good one.
Right.
And then you get the DNA sequence, and at this point, the way most of these drugs are made is that once you have that DNA sequence, you make a special cell that can produce, it's actually hamster-derived cell, that they can grow in these 2,000 liter bioreactor vats. They look like big beer vats. And they just grow 2,000 liters at a time, and they pull out the antibody and purify it. They test to make sure it's pure, and then that becomes a drug.
How do they shift it so that it goes from mouse or hamster to human, so that there isn't that kind of I know we go into the weeds here, but I think it's important. People are so frightened of everything right now. Maybe if they knew more of how it's built.
Sure. So the technology is called humanization. And what they do is the mouse sequence for an antibody looks different enough from a human sequence than if you inject it into someone. They'll get they could get serum sickness and anaphylaxis or some other negative
really
responses.
Bad, yeah.
The other thing is their immune system will sort of reject it like a graft. So you'll start making antibodies against the drug, and the drug doesn't work anymore. To avoid that, what they realized is, well, the general shape of an antibody is about the same between a mouse and a human. So I bet we could just basically clip out the parts of the antibody that binds to whatever its target is, and we can graft those onto a human antibody. And so then you have an antibody that's almost entirely human. There's just these little loops that bind to your target that are mouse-derived.
And the loops are the antibody?
They're like the fingers of the antibody that binds the target. So you take the rest of the antibody, which is really the chassis, and you take the little fingers that make it specific, and you transfer those over to a human antibody. And then the body tolerates it much better.
Okay.
So that's one way. The other way that people did it is they said, well, we should get totally human antibodies, not even use a mouse. And so the other technology is that you can take blood from a whole bunch of human donors, and there's a way to take all the DNA from all their B cells to make all their antibodies, distill it down into something called a library.
Do you combine them?
You
like
combine
the
them,
O, the A, B, all of those?
All those people.
All those bloods. Okay.
Yeah, and you take just the DNA that makes antibodies, and you isolate it, and you make something called an antibody library. And then you're not using immunization anymore. You're using robots to go dip your target into these, let's say there's 10 billion antibodies, and you pull out the ones that bind to it. And that way you get a fully human antibody. And so when I was working there, both of these technologies were being used, and they both had issues. And I came in with kind of the guy who hired me was, in retrospect, very helpful, where he's like, right, so your job is to make that guy happy, therefore make me happy. I'll see you in 6 weeks. And he left. And so instead of being told you're going to make an app, right, or you're going to make something where my powers would be limited by that person's perception of an imperfect understanding of what I could do, the goal was, and like,
happy is a little vague too.
Yeah. And basically, like, satisfy the site lead was the goal, which is like that's an awesome mission, right? Because then I went around the building, and I'm like, all right, what can I do that could have the most useful impact on this organization? And it also opened up doors, because the group was doing everything from early target discovery, trying to figure out what you want to go after, all the way through the biology of proving it, the engineering of making the molecules, everything up to clinical. And so I could go knock on doors and try to understand what people are doing. And if I just wandered in and was like, hey, what are you guys up to? Teach me everything you know. They'd be like, hey, I'm a bit busy. Could you bug off? But because I came in with a computational skill set, and I was like, look, I'm trying to find ways. To tell me what you're doing. I'm going to try to see if there's ways I can make your life easier. Then they would open the doors for me.
See, that's really sell them what you have that they need.
Yeah. Right. And then I benefited from like an immense amount of training from experts, and I exchanged the favor by building tools that were useful to them. And the most useful tools that I built it's really a technique I learned from Kim and Shalander. It's an algorithm called a hidden Markov model. It's the same algorithm you have in your phone that turns your voice to text. They're starting to use different ones now, but it's a popular one. It turns out that algorithm is really good at interpreting antibodies in an efficient way, because they mutate and they change. So they're kind of hard to see, just like there's lots of people who have different accents for saying the same word. The hidden Markov model algorithm is very good at fitting and understanding the pieces of the antibody. And so I built tools to help the hybridoma group and the antibody library group enhance their discovery process to be able to search through more antibodies faster. And when I built these tools, they didn't really have good computational infrastructure, and so I had this little Lenovo laptop that they gave me, and I reformatted this Linux on it.
I'm picturing this.
It was like drove me nuts, honestly. But I was like, this laptop's so slow, but Linux is very efficient. So I reformatted it as Linux, and I got tired of people coming in my office because I built these tools and like, hey, could you run it again? Could you run it again? And I was like, oh man, this sucks. And my colleague I shared an office with, he was like, Jake, what is the problem? Now you have the job security. And I'm like, no, this means I can never get promoted because I always have to
you're doing these accents. Thank you.
So my buddy
We've got
Andrea.
British and French so far.
Yeah.
So I'm picturing who you're working with now.
Yeah. And so I was like, I'm going to make a little web server. So, and I checked and no one, I wanted people to be able to access it without having to come in my office.
Right.
And so I made a little web server. They could click a button and run the different tools.
Right.
And then I called Pfizer because my laptop was called like AMR 947 blah, 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 .pfizer.com. And I was like, can I give it a DNS name? And they're like, no one ever does that, but okay. And I was like, oh, really? I'm like, is antibody.pfizer.com available? And it was. And so my little laptop was the server, antibody.pfizer.com. And within a couple of weeks, the people all over the site were using it, and I could build new tools because I wasn't having to run each one for them.
That makes so much sense that you don't have to keep serving them one at a time.
Yeah.
They can.
And I was collecting the data too, which was really helpful because for future use. And then suddenly I noticed my laptop started like just purring all the time. And what happened was the sites had kind of met around some of these technologies, and now my laptop was being used by maybe 8 sites. Oh. And so I was collecting data across all the organization at Pfizer with this like it would have like absolutely driven IT crazy if they realized that was a major server. and the timing was just coincidental that there'd been this arms race for like the faster, cheaper genome sequencer that, we got the genome out in 2000, and then now we're talking, this is 2008, and now they had a device that could sit on a benchtop. And the site I was at had gotten one for other reasons, but I went over to that group and I said, hey, look, I know this is weird, but I was wondering if I could just I don't want to look at the whole genome. I just want to look at this huge diversity of antibodies, because your genome has 26,000 genes, but your antibodies have maybe 50 million different unique antibodies. So it's actually a bigger problem than the total set of genes through the shuffling. And I was like, by my back of the envelope calculation, it does not make sense to me. The math doesn't line up on why we're not getting certain types of antibodies from the mouse immunizations or from the antibody library. I want to use this new instrument to look really deeply in the immune system of these animals and of these libraries to understand what's missing. Mm-hmm. And so they were like, all right, that's kind of crazy, but sure, let's do it. And I had support by, I think a couple of our leadership were super leaned in on this idea.
Good.
And so I was supported by them, which was great. And so we did what became one of the pioneering studies of this whole thing called repertoire sequencing, which is using these genomic sequencers to look deeply in the immune system. And it was like stunningly groundbreaking because it suddenly let us open the hood of the immune system and try to understand what's going on in this otherwise very complex black box. Because that shuffling diversity lets us fight pathogens. It also made it really hard to interpret what the immune system was doing.
Because it's always moving.
And it's so diverse. And it's always moving. And so this suddenly gave me a tool to be able to take these super deep snapshots of tens of thousands, hundreds, even millions of unique antibodies all at the same time. And when I did it, Pfizer was letting me publish. So I published a series of papers. And I was telling them, look, this immune system is not doing what Janeway, the immunology textbook, is describing. There's differences. And I started getting these ideas as I was doing this, first on how to improve the antibody libraries. And I did a lot of work using DNA synthesis for that. But my deeper, and ultimately I think for my career, more meaningful thing was I started realizing that people were making these incorrect assumptions about the diversity dynamics, this machine of the immune system and how it responds to proteins and pathogens. And I had this idea on why I was realizing is why people were describing why the immune system misses certain key sites on viruses.
So in other words, why people get sick. They were misinterpreting the reason behind that.
In particular, I think why it is that there are these kind of Achilles heels, these conserved sites on viruses. Flu has them, coronavirus has them, HIV has them. Why is the immune system misses them? Like, why, if they're always there and conserved, why does the immune system target these sites that are so easily changed?
Right.
And there were some like kind of goofy theories that existed in absence of this data. And suddenly I was looking at this data that told me, no, there's another explanation. And the reason that was profound is it gave me an idea on how to fix it. And this is an important thing. Since 1942, I believe, is the first time we created a flu shot. And then by 1947, they had stopped working and people realized it's because the virus had mutated. And ever since that time, we have been stuck in the Sisyphusian challenge of making new vaccines
and
for
hoping
flu.
It's the right one.
Yeah, and having to keep update them and hoping they're not too mismatched. And we're still, this last year, we didn't do a good job.
Well, that's how I got the flu, as a matter of fact.
Yeah, you could get vaccinated. The Southern Hemisphere, 34.5% efficacy, which means about 1 in 3 people who got a flu shot benefited from it, and 2 out of 3 could have gotten vaccinated and still got sick. And we all experienced that from the coronavirus vaccines. They looked great when they first came out, but as the virus started mutating more, then you'd still get sick. And the argument was you should still take it to avoid serious disease, but it was by no means a guarantee.
Well, let's save we haven't gotten to COVID yet.
Sure.
We'll get to COVID really soon. But this is just fascinating. So after that was published, then. So
I published four papers there, I think. And I developed methods for looking at the immune system and then also using DNA synthesis to build better, like to fix the things I saw as defects in these antibody libraries, to build more powerful synthetic immune systems. And I think that activity of observing and building, because there's nothing quite like building to test your theories.
Right, because it's not just talking about it or drawing it.
Yeah.
You have to actually make it.
I think if you look at science, a lot of breakthroughs happen from people who built something, because it's not just good enough to be statistically effective. Your theory has to work well enough that the engine runs.
It has to actually live in the physical world.
Yeah.
Yeah. And
so I had cycles of that. And then by 2012, I had been promoted 4 years in a row, and I had a BA at the time. And so I was a principal scientist with a BA, and I had a lot of people being like, Jake, why do you want to do anything else? You have job security. But the problem was I had started having these like really big ideas, and I recognized they were risky and they could be wrong, but they were so big I couldn't walk away from it. And so I resigned from Pfizer. I started my first company, Distributed Bio. And I also applied for the Stanford PhD program in computational and systems immunology, which is this new subdiscipline of immunology. And I looked at it again
more slowly.
It's computational and systems immunology.
Okay.
It was what I was doing. They were just given a name to it.
Right.
And there was this guy, Mark Davis, whose work I had read quite a bit. And my grandfather's advice to me was always, you go to work with the person. It's not so much the institution, it's the person. That's what's going to drive you forward in a new field. You
come from great wisdom genetically, even if we don't know about your immune system, but these people are very wise. Your grandfather, your grandmother.
I owe a lot to them, yeah.
Yeah.
And so I applied. I did both because I wasn't sure if I was going to get in that program. And I also am starting a new venture. You're not sure if it always works out. So I kind of just did both, and they both did. So I didn't sleep a lot for the next 5 years. But my first company, Distributed Bio, we worked on using, again, these computational optimized antibody libraries, and then also licensing access to antibody discovery, to Amazon cloud-based software tools, using those algorithms to analyze the immune system. And then ultimately we expanded out to services. And we were very successful.
You did good. That's when I first met you, when you came into my studio.
That's right.
Yeah.
You
were with my business card that I have of yours says Distributed Bio.
That's right. I was still the CEO of Distributed Bio.
And then you sold it.
I did.
You sold the company, which was wonderful to hear about.
So we built it up, and in Distributed Bio, that's when I started running these studies of this universal vaccine concept. And so I incubated it at Distributed Bio. One of my co-founders basically thought it was a boondoggle, but I was making enough money with other things. He put up with it. I think my other co-founder saw it could, we all knew it was a long shot, but it could work.
Universal vaccine.
Yeah.
So when I was the CEO myself of San Francisco Media Group, we had different physicians panels come and speak down on our stage. And I mentioned you and the universal vaccine to some doctor who laughed and laughed and said it was impossible.
Yeah.
And I just didn't say anything back because I was like, I don't believe in impossible. So How is that research going now?
Yeah, so we have our clinical candidate, which is a pan-influenza universal flu shot. It is in manufacturing for human trials right now. So we're less than 9 months from human trials. The clinical candidate, it can protect, neutralize, do all the things you want 13 years in the future beyond any of the components. We did a study where we only used components up to 2007, and then we challenged ferrets and pigs 13 years in the future. That was supported by the Gates Foundation and this thing called a Grand Challenge.
So when I first learned about your work was in the Netflix series Pandemic. And I didn't watch that until after we were in a pandemic, because up until then, I think a lot of us, including myself, scoffed at the idea that there would ever be a global pandemic. So I remember seeing the series advertised and going, up, I don't need to watch that. But after I was, dealing with COVID-19, it was probably a couple months in, and I had met you in my studio. I was like, okay, I got to watch this.
Yeah.
And so you had a flock of pigs down in Guatemala. Is that who you were testing on?
That was my test. Yeah. So I'm an affiliate professor of the University of San Carlos, which is the public university down in Guatemala. And I had this phenomenal, I still have a phenomenal colleague named Erwin Calgua, who's a professor down there of epidemiology. And I had this idea of this epitope focusing. Back in 2012, when I first started developing this, in 2015, when we started doing the studies, it was reasonably far out to talk about doing a universal vaccine. I think that's a pretty moving target right now. We have other companies working on efforts. There's obviously a huge need and recognition.
Are there other companies now? Yeah, and it's recognized as something that's a possibility now.
I think there were people working on it, even a couple people back in 2012, 2015, but now there's like multiple active organizations. The presidents have had three. I guess Trump in his term, and then Biden, and now Trump presumably again, specifically called out funding and resources to be able to enhance broad spectrum or universal vaccines.
Right.
I think the nature of new technologies is that everyone says they're impossible until they're done, and they just become the way. So
that's starting with the horseless carriage, et cetera.
Starting with fire. At some point, someone had to pick up the fire, and someone said, "We've never done that. We've never touched it. Only bad can come of it." And then someone's like, "I'm going to try it anyway." And then we cooked food. So there's multiple organizations. At the time, it was early. I was unable to fundraise from it in the beginning because first off, I didn't have my PhD in immunology from Stanford. I think that's one of the reasons I did the PhD at the same time, is so that if I was wrong, I wanted to be surrounded with immunologists to punch a hole in the idea so I could do something else with my life.
So that they were, smart enough and knowledgeable enough to really help you. And if I was, that makes more sense. That's good.
And if I was right, I needed to being a card-carrying immunologist from a premier university so people could hear me.
Yeah. And get the support of colleagues who were smart.
And so we ran those studies in Guatemala. We did, I think I was able to do like 5 cycles of studies and the first time, you're setting it up, there was challenges on moving samples back and forth because it was supposed to be easy. And then because of Ebola, they changed all the rules on serum shipment. And so there was a long period where we're running a study. We just had a bunch of pigs. We were immunizing them with this vaccine and then waiting. Bringing the vaccines down was not that much of a problem, but it was getting the serum back to our laboratories in the United States because my facility in Guatemala was very simple.
Oh, yeah.
Where we were stuck for many months. And I wasn't sure, am I just, you're shoveling manure in Guatemala and you're like, am I just wasting my life? What am I doing?
The glamour of working with the pigs.
Yeah.
Right.
But also I feel like if it doesn't feel weird, then it would just be the way and it would have been done already.
When you said the people who said, "Oh, that's kind of crazy, but we'll try it anyway," I thought, crazy is often the way to go.
Yeah.
Because that means you've stepped outside of what's the normal box that everybody does their functioning within.
And if you want to build something new, if you want to change the way things are, you have to do something unusual, because if you follow what everyone else is doing, then you're going to get right back to where we're currently
right.
are.
Then it doesn't change. So okay.
So we ran the studies, and then they were working. They looked phenomenal. We ran these optimization cycles. And it was actually, I think at the end of that, they were filming that in 2019. So it was filmed before the pandemic struck.
Yeah.
And right at the end of it, I think they filmed the part where we end up getting the Gates Grand Challenge grant. And
I think so. Yeah. I remember that part.
So we ran that. The studies were run during the pandemic. And we saw this like astonishing ability of the vaccine to protect like 13 years in the future, even with a pandemic shift, the swine flu. We chose components only up to 2007. So we didn't even include components from swine flu and beyond. And yet the ferrets and the pigs were protected.
There were ferrets too.
Ferrets and pigs, that's right. Out to...
And let me just ask, why did you choose ferrets and pigs?
Yeah, so I chose pigs because...
They're similar.
They get flu. They're actually the biggest source of pandemic. We've had 5 pandemics of flu in the last 100 years, and they often happen because pigs get infected with bird flus easier than humans do, and they have pig flus, and they can also get infected with human flus.
Right.
And when you have a pig that's infected with multiple flus at the same time, the genetic material can shuffle, and that shuffling sometimes can create a pandemic strain. And so the other reason was pigs have a flu that's much more similar to humans.
Right.
Like we do mice work, but in general, I don't like the mouse model of infection because
it's
the mouse
too far away from humans. And the ferrets?
So the reason you do ferrets are that for historical reasons, people have claimed, which I kind of agree with, and I kind of think it's just historical precedent, but they claim that the lung tissue is very similar in ferrets. And so for historical reasons, for decades, the ferret is the go-to animal to test flu shots. And so if you want to compare your flu shot to all the other historical flu shots, you go through ferrets.
Yeah. Do you feel like there's some reason why there are more pandemics now? Is it just a larger population? Is it how close many people are living with animals? What would you say? Because this is just kind of feels like we're moving along pretty fast now.
There's two parts to that. One is, the rate, and I'll explain the reasons that as much as I know them, to why I think we are in an accelerated period of global outbreaks and pandemics. But the other one is human memory. The pace of pandemics, they've been happening to us, major outbreaks and pandemics, since the beginning of time. We notice them and we forget about them because they happen just every few years, just outside of our memory. But if you think about what's happened to us in the last 100 years, we've had 5 flu pandemics, including one that killed 50 million people in 1918. We've had HIV pandemic and now endemic. We've had 2 different coronavirus outbreaks, one of which became pandemic.
Which was the first corona?
There was SARS. SARS was a coronavirus.
SARS, that's right.
It was 2004, 2006, that period. We have multiple Ebola outbreaks through remarkable heroic effort by the teams in Africa. They've largely kept it in Africa.
Yeah.
And then there's been a bunch of other things. We had H3N2 came into the human population in 1968. We didn't used to have.
Which one is that?
That's one of the 3 flu strains you get vaccinated against. It didn't used to be in humans, and it came in 1968. So we actually are getting bombarded with, there's also one of my kids got something I'd never heard of before. It was like hand, foot, and mouth, or it sounds like the animal thing, but it's different.
We used to have, I remember that, like they used to call it hand, foot, and mouth disease or something.
Okay.
Yeah.
So something I didn't remember
That's from when I was a kid. I remember hearing about
so that
it.
has been around. Okay, I'd
In
never
other words,
heard of it.
Another generation older than you, we had, and my dad just shortened it to hoof and mouth disease.
Nice.
Hoof and mouth disease.
So there are these expanded, there has been this history, and it goes back for centuries. There's actually this fascinating literature. It's one of my hobbies, is to go back and look at the history of pandemics and outbreaks.
That's really smart. Like the bubonic plague and Black Death and those kinds of things.
And so there has been this history. It's actually been going on for a long time. I mean, polio, we used to have horrible outbreaks.
Right.
And then there's things like smallpox that was a scourge of humanity back to the pharaohs. And yet we were able to eradicate it in the 1970s. And so there's this history we've had, I think, where there's been an immense amount of constant infection harassing humanity. We're actually in a position where there has been improvements. We got a little bit of a reprieve because of the vaccine era. But to answer your question, yes, we're in a period where there's more infection than ever. And the reason is, or the capacity for global outbreaks, the reason is we have a lot more people, is one. The second is that people travel a lot more than they used to.
Internationally.
Yeah. When they used to be limited by boats, there was a certain amount of essentially built-in quarantine that would happen where an infection could burn out in a boat before it reached its destination over a large ocean. And that's not true in planes. And then the third issue is that many of the diseases that we get infected with also infect our livestock. And so the current H5N1 outbreak is an example of this.
The bird flu.
Yeah, we have 8 billion people on the planet, which means we now have these mega farms of cattle and they're near birds. And so for the case of H5N1, which we call bird flu, but it's really now like cattle and spillover into human flu.
Right.
What happens is the birds bring it into the poultry farms through wild contact. And then because you have a huge amount of poultry that are near pigs or cattle, the more animals you have adjacent to each other, the greater the chance of spillover happening. And when spillover happens, it's like what we're seeing in humans right now. At first, there were some cows infected and it didn't transmit very efficiently, but then it adapted. And so now it's transmitting between the cows like wildfire.
Right.
And that means that you now have a whole bunch of cows that are near a whole bunch of people. And this is a new phenomenon. We did not used to have these mega factory
farms. Big farms.
And I saw a test one time where they found 73 different mutant versions of influenza on this one mega farm, and they're shuffling. And so there's it's like the more animals you have, the more you're playing that little slot machine. Every time a new animal gets infected, that there's a dangerous mutation.
That it could shuffle and change. What was different about the COVID-19 vaccine, I should say vaccines, than other vaccines? That why some people became ill from the vaccine itself? Some people actually developed big life-changing symptoms from the vaccine.
Yeah, sure. So,
and I don't believe that that's just hearsay. I actually do believe that there are people who have suffered from that.
I think it's important when we talk about vaccines to be forthright about how effective they are, but also not oversell it, and also not to sugarcoat the side effects, which they do have. I think there's this urge by epidemiologists to give no ground, because there's been some false information around side effects that doesn't appear supported by science. But I think that gives rise to this effort being let's just say there's no side effects because we don't want to confuse the population. And I don't think that's fair either.
That's not fair either. No, that's not truthful either.
So I think when you do that, first off, people are smart. And the epidemiologists are just give them a super clear message so they'll all take the vaccine. And I'm like, no, man, if you do that, then they're just going to be I don't know what's going on, but I know I'm not getting the full story. And you're just going to... you're better off sharing all the details, which is what I tried to consistently do through the pandemic. So with respect to the vaccines that were created, there were different vaccines and they had different efficacy profiles. That's how well they work. And then they also have different safety profiles. So I'll speak largely about the mRNA vaccines that I think had pretty comparable profiles, separate from J-Covidin, for instance, which was actually a different virus and they put a spike on it. So it's a vectored vaccine. So for the mRNA vaccines...
mRNA.
The mRNA vaccines. mRNA.
What does that stand for?
mRNA is... Oh, God. It's messenger ribonucleic acid. So you have DNA. That's your hard copy in your library. And and that's stored in the nucleus of each cell. And when the DNA... the DNA instructions need to be used to build something. Rather than taking the book out of the library, you make a copy of it. So if they want to build something, they make a copy of the master book, which is the DNA, and they copy it into RNA and mRNA. And that goes out of the cell, and that doesn't last long. That transcript may last a few days, but it's enough to make some copies of whatever the instructions are to make. Mm-hmm. And so it turns out to be an attractive thing for delivering a vaccine, because the old ways we used to make vaccines back when Edward Jenner first did it is you'd take a virus that was a weakened cousin of the pathogen. So instead of smallpox, which would kill people, you would take cowpox, which was
This is what I remember. Or they would say things like, there isn't a live cell, or there isn't any, that kind of.
And so that was a live virus that they would give. Those are called attenuated. And people had made different vaccines in the olden days that actually worked pretty well because it was weak enough you could beat it, but it was similar enough that your antibodies and T cells would cross. And then as the technologies advanced, they started making these things called subunit vaccines. And they said, instead of delivering the whole pathogen, maybe there's just a spike or just one part of the pathogen that you really want to target. And then we can just deliver that as protein and the immune system will respond to it. And that'll be enough for it to fight the rest of the pathogen. That's how flu shots work. They typically take out the hemagglutinin and neuraminidase, the two proteins on the outside of the virus, and they get rid of all the rest of the virus. So the vaccine can't infect you.
Can't give you the flu.
Yeah, it just gives you these proteins. And so the RNA technology, the idea was instead of producing the proteins, which the trick with protein, any protein you put in your fridge, eventually it will go bad. And they do a lot of tricks to make it last as long as possible, sort of spamize it. But eventually you have issues with proteins. You also, if you're injecting protein, it's not as good of a mimic of being infected as this idea, well, what if we inject the RNA? And the RNA will get into your cells and it'll make your cells make viral proteins. And then your immune system will see the protein, but you also will think that that cell's been infected and start attacking it and produce a T cell memory, a more effective immune memory, because it more effectively mimics an infection. And so the technology had been worked on for years leading up to the pandemic. It had a couple notable advantages, and I'll talk about the disadvantages as well. So the advantages were it was super fast to be able to manufacture because it's a lot simpler to make.
And it was an emergency.
Yeah. And so they did it in, I think, 43 days. They did the GMP, that's a manufacturing thing, which is astonishing compared to proteins where you're talking about, I mean, typically it's a year and a half. You can do it faster in a pandemic, but it still takes a long time because you have to make special cell lines.
Right.
And then the other advantages were that it was very potent. So it turns out getting your immune system to think that your cells are producing viral proteins, but without producing all the pieces of the virus that make it infectious.
Right.
Is good. You get very potent responses and you get these good antibody, but also T cell responses, which are important, two arms of the immune system. So those are the good things. And when the vaccines first came out, they looked amazingly effective.
The COVID vaccine.
Yeah, the RNA vaccines, they were like 94% effective. But that's also because the pandemic had just started. And so the virus hadn't had enough time to mutate.
It was just sort of one kind of virus mostly.
And so it looked great. That's the whole point. These vaccines work great if you can target something that looks like the virus. But once it mutated and differentiated, especially when Omicron came along, the response dropped. But there were side effects. The mild but annoying side effects were, in general, the more effective a vaccine is, it can give rise to more headaches and sore arm and body fatigue, maybe some shivers, and some of that stuff can be mitigated. Some of that stuff is fundamentally
and it passes too, usually. Yeah.
And so that stuff, look, these were potent vaccines. They also were, I think both companies, Pfizer and Moderna, were like, we need to give enough to make sure that this is effective. And so they erred on the side of giving higher doses, Moderna in particular. Which I don't necessarily think was a bad strategy in the circumstance, but it did give rise to more side effects.
Bigger side effects.
In peacetime, they might have tried a little bit of a lower dose outside of a pandemic. But there were some, in most cases, rare, more severe side effects. And I say in most cases because there was a unique phenomenon with boys aged 12 to 17 where it wasn't that rare. And the side effect for mRNA was this issue that it was 1 in 100,000 subjects or so of most ages would get myocarditis or pericarditis. And that's an inflammation of the heart.
Inflammation of the heart lining, right?
Yeah. But in boys aged 12 to 17, the rate was much, much higher. And it was more like one paper said 1 in 5,000, another one said 1 in 20,000.
Wow, that's pretty high.
And let's average them, 1 in 10,000.
Yeah.
But and that's defined as severe enough heart inflammation that they had to go to a hospital and get successfully diagnosed. Now, 2/3 of them resolved within a few days and they were okay, but a third had more extended complications.
Right.
And look, I'm a vaccine scientist. I work on these things, but my feeling is that I think I know some boys age 12 to 17. I think some of them could get very sick before they tell someone, or it's not clear that they're not just hiding in their room.
Right.
Furthermore, you need to have someone who takes them into a hospital, and then the doctor needs to notice and then needs to correlate it to the vaccine.
So there may have been a delay before they got treated.
I also think this could have been a higher rate than is evident. Like if you go 1 in 10,000, that's the subset of a subset that made it to a hospital and got associated.
It could be
and so I think
more.
it could have been a bigger problem.
What was different in the structure of that vaccine that made the side effects so severe in some people?
So this is something I work on a lot because I think, I grew up with hippie parents. I'm a vaccinologist, but I also I think I push harder than is average for vaccine research to try to make them as safe as possible and not just be like, well, the rates are acceptable. And so my team has looked at this a lot, and there's a couple theories of what was going on. I do not know why it was boys and not girls. It was something about the post-puberty, maybe it was blood flow, maybe something else, maybe they exercise a lot. It's not clear why. There's something clearly about, I would guess, new testosterone.
Something about their gender and their hormones and whatever.
But the theories on why was that happening, I have a couple different theories. One of them was the LMPs themselves were somehow inflaming the heart. mRNA LNP vaccines, they are the mRNA, which is the genetic information that encodes the spike of the coronavirus. And then you wrap it up, because RNA gets broken down very quickly.
So
to
wrap
protect
it
it, you wrap it up in cholesterol and a couple other chemicals. And that's called the LNP, lipid nanoparticle.
And somehow that might have been inflaming the heart.
That was one theory that maybe I was always somewhat skeptical of that theory, because you're putting in such a tiny, you're putting in like 30 micrograms, 30 millionths of a gram of this stuff. It's possible that some of it, and it goes in the arm, it's possible some of it could have drained into the lymph node and then got into the circulation.
Well, that's interesting. I wonder if it made any difference if they were vaccinated on the left side or the right side, or if closer to the heart or further.
That's an interesting idea.
Further from the heart?
Yeah.
I never thought of that either. But
so it could be. I think my other theory is that these things, whatever cell absorbs them, is then going to start producing a viral protein.
Right.
Now, they're designed to get into the muscle of your arm and then go into the draining lymph node and produce. So the spike protein should be produced in a bunch of muscle cells, but also produced in immune cells in the lymph node. And we know that the lymph node would swell up afterwards and produce that strong response, which is expected. But I always wondered whether, okay, maybe for some reason in boys it's happening easier, but what if some of that stuff is draining out and it's getting into the circulation? Then some of those little LMPs are going to fuse with heart tissue and cardiovascular tissue.
Because it goes into the bloodstream rather than just hanging out in the lymph
if it doesn't get absorbed, it somehow gets in the bloodstream, and then it ends up in heart tissue. And then you're going to have your heart cells are going to start producing a viral protein. And your immune system is going to think that that's your heart's infected. And so you're going to get heart inflammation because of immune attack.
Right.
And so that was one of my... I still think that that's probably the most likely explanation. And we ran a program at Synovax to try to create RNAs where we put this, there's a little kill switch called a microRNA that you can put on an RNA that says if this RNA ends up in heart tissue or cardiovascular tissue, kill the RNA right away.
And does it work?
So where we got to was we have the elements in there. They look like they have differential expression, but what we wanted to do was test it on an animal model of myocarditis, and there was a published animal model, and when we tested it, the published animal model didn't work. So the group that reported that they could induce myocarditis was unable to do so.
Right.
And so we have a technology that could be applicable here, but we don't have a way to prove it before, and it's kind of irresponsible just to try to we think we're smart, so we're just going to put it in humans without testing. So we're kind of waiting for a better model to test that. But that was my one strategy, was maybe we can inactivate it so that if it gets in myocarditis tissue, it shuts down. And I think that makes a safer vaccine. The other thing we're doing is we're looking at lower doses.
That's what I was wondering when you said that maybe it was overkill. Maybe they made it too high.
Yeah.
Maybe they talk about microdosing of other things. Maybe small doses.
Yeah, and
so the body doesn't go into kind of a shock state.
And that's kind of how our technology fundamentally works. I spent a lot of time on enhancing the behavior of our RNA so it's extremely potent, so we can put much less of it in, and therefore there's less LNP. But I have this guy, Gerald Sadoff, who joined as our chief medical officer, and he's gotten 14 vaccines approved, including Gardasil, MMRV, the Rotovec, the Jacobden from J&J during the pandemic, and 14 vaccines in total. And he came in with a couple engineering techniques that we applied to enhance the potency of our proteins and make them last longer. So therefore you can deliver much less RNA.
Much less of the,
And the last thing that I've looked at we're doing collaborations with a couple companies that have these patches. So instead of injecting 0.5 milliliter of vaccine into the arm, you put a patch on the skin and it basically just delivers it to the top layer of skin. And so that would, if it's an issue of either the LNPs or the RNA draining to the heart, that would kind of get rid of that.
That would take care of that. What is it about COVID-19 that made it so hard to chase, basically? I mean, it replicated so fast over and over again.
You're reminding me, and I'll answer that question, but there was one final reason why the coronavirus vaccines could have had a unique problem.
Yeah.
And that is that the spike protein of the coronaviruses bind with extreme tightness, affinity, to something called ACE2. That's the receptor. And that ACE2 is heavily decorated all across heart tissue.
Oh.
And so the other issue is that this could have been a unique problem for coronavirus
Because of the
vaccines.
shape of how many protein spikes there were.
Yeah, those protein spikes bind to heart tissue. And so you could either have these things expressed in heart tissue, or it could have drained in the lymph node, and then you have immune cells that are suddenly expressing a viral spike that then cause them to become stuck like Velcro, or like those little burrs you get when you go hiking.
Yeah.
Those cells would have gotten stuck all over your heart tissue.
Ugh.
And so that could have been this could have been a uniquely coronavirus problem. So those are the theory, and our solution there is we have mutations that remove that stickiness to heart tissue, to the ACE2 receptor, because you don't really need that for the vaccine to be effective. They just moved quickly and they didn't change it, which is unusual. And in our assessment, I think that was a little dangerous. To get back to your question,
just curious about it, because it seemed like there's this and now there's this, and Omicron, and now it's boom, 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 and there's a new mutation, and
this is the big problem, but this is also where I think I have some optimism towards the future. So the coronavirus, it's in good company. It mutates rapidly, and it makes us update our vaccines. And so they've just updated a new one a few months ago, the JN1. I mean, I work in this space, and I get tired of having to track all these changes and strains. It's annoying. And the reason we track them is that our vaccines we currently make, and really the technology we've had since Edward Jenner 200 years ago, has been to make a vaccine against a single strain and then to use that until that strain looks too different from the mutant forms and the vaccine is no longer useful.
Right.
There are certain pathogens where they just don't mutate that much. And Edward Jenner's technology and things that came after were good enough to eradicate smallpox and drive polio down to near eradication, dramatically reduce measles, mumps, and rubella, and so forth. But there are pathogens that mutate just fast enough that the old technique doesn't work very well. And coronavirus is in good company with flu, where we also update the vaccine 2 times a year.
Every couple
and even
years.
then it doesn't work that well.
They do
it twice a year.
Yeah.
The northern and the southern hemisphere have 2 different flu seasons. They're 6 months off.
That makes sense, because of the weather changes, And
then they have, we've never made a working HIV vaccine because it mutates quite quickly. And there's a bunch of other pathogens. Like Ebola, they have one vaccine for one.
Just recently, right?
Yeah. Mm-hmm. They're working on a second one for another strain. There's going to be another strain that pops up, and we'll have to make another vaccine. Marburg is related to Ebola, and they don't have a vaccine.
And what you're doing, though, is to try to create something, and I don't know how you're going to do it, universal vaccine.
Let me tell you how I do it.
Please tell me, because I still remember that doctor scoffing at me when I mentioned that to her.
First off, I'll say it's useful to have a diversity of opinions. So I think that's fine for people to have different perspective on these things. And it's my job to shift people's opinions with technology. So the thing with all of these viruses, like if I go back to that story of flu, by 1949, they realized that their vaccine they just made, and everyone was so excited. We finally have a flu vaccine. This is a really big deal. They just had a couple, a major pandemic in 1918 that killed more people than all of World War I. And then they'd had other major outbreaks, and they finally had a vaccine. And then suddenly a couple years go by and it doesn't work anymore. And they had relatively primitive tools back then, but they were able to determine that the serum of vaccinees was not crossing to the new virus. And they realized they had to update it. And so they were in this process then of updating every year and occasionally adding new strains like H3N2s. And I think that for a while people thought that's just your tough luck. That's just what we're always going to have to do.
That's what we've always felt. Yeah. I mean, we've grown up going, we need another flu shot this year because it's a different strain.
But the thing that happened that changed people's opinions about this was in 1993, there was a group that said, hey, you know what? I've been looking at a lot of people's serum, and it turns out there's this one subject that seems like they're producing serum that's crossing not just to one strain, but to a whole bunch of strains. And as the new strains come out, they're crossing to it even though they're new strains. And they went and they isolated individual antibodies that this person was making, and they realized there are these antibodies, individual antibodies that are crossing lots of different strains. And they said that means there must be some conserved site that the virus isn't able to mutate across the strains. And then in the 2000s, like 2000, 2004, 2006, there was this kind of golden age of these papers being published where they were crystallizing. They were using a particle accelerator and a lot of X-ray to be able to figure out the exact location that an antibody would bind to the hemagglutinin and the spike on flu. And they realized that and they started sequencing a lot of flu. And they realized that flu has this Achilles heel. There's really 2 sites. There's 2 sites, even though it can mutate most of that spike, there's 2 spots that it can never mutate. And the reason it can't mutate them is that if it mutates that site, it's no longer infectious. And So these sites are these little...
So those are the vulnerabilities.
Yeah, and they're always there. And the astonishing thing is like, as soon as people saw that, they're like, if we can just get everybody to focus the immune response against those sites, we'll have a universal vaccine. The challenge was how to get the immune system to focus on it, because most of the surface changed and the immune system didn't know to focus on those sites over all the other sites. And people came up, and by the way, they started finding that this was also true in HIV. HIV has multiple conserved sites that cannot mutate. And the main reason, if you think about it, a virus has to infect one of your cells. And in order to do that, it has to dock up against a receptor. And so imagine it's like a spaceship. It can't mutate like the docking seal. Otherwise, it will no longer be able to dock.
So that's the docking seal.
And so, it's conserved. It has to be unchanged over time. Otherwise, the virus can no longer infect.
Because the cells aren't changing. Only the virus is changing.
Exactly. And if you can get an antibody against the docking seal, the thing can't dock either. So it's a perfect target. It's just getting the immune system to focus was the challenge. And people came up with a couple hypotheses on what was going on, and they tried some of these techniques and they weren't working. And that's really it was in that context where I started doing that work at Pfizer with the deep sequencing and my kind of combination of population genetics, deep sequencing, and structural biology, where I was looking at these systems and realizing some of the theories on why they would say that these sites were not immunodominant, which just means that the immune system misses them. And they had weird theories for why they were being missed that didn't make sense to me. And my answer here was I was like, well, no, I think the problem is just that there's like hundreds of millions of ways an antibody could bind the surface. There's so many different ways, like slightly tilting and different angles and different spots that antibodies could bind. And of all those millions of ways, it was really only a few thousand that were just on the right spot. And so the problem was just numbers, that you were throwing darts at a huge dartboard with a tiny little red bullseye. And so if without something to focus them, you were going to miss almost all the time.
Right.
And then that's what also got me interested in my technology solution, where I was like, some of these other things are never going to work based on my understanding of how the system works. And so I thought to myself, what if I were to use evolution against itself here? What if I were to take 100 years, sample like 20 different flu strains from the last 100 years, where it's mutated for a century, and it's mutated just about everywhere it can, but those two sites have not mutated. All these 20 components share those sites. They're identical. And then what if I mix all 20 together and I dilute the mixture so that there's not enough of any one of the individual strains, there's not enough dose to produce a strain-specific response. There's not enough of that component. Then if I inject that, all the B cells that only recognize a single strain, there's not enough vaccine there for them to respond for their one strain. But pumping through our veins, there are these rare but powerful B cells that would recognize that conserved site. And if they recognize that conserved site, then from their perspective, all 20 of those things are identical.
And the conserved site is the Achilles tendon you talked about.
The Achilles heel, that's
Achilles
right.
heel.
Yeah, there's two on flu. There's one that it needs to bind to the surface, and there's another one where the spike changes.
Always the same on all of them.
Yeah, they're super conserved.
So that when you combine them all, synthesize that, put it all together, you've got those sites.
That site's like 20 times higher concentration, 20 times higher dose of that site, because they all share that site. They're different everywhere else, but
so
they all have that site.
So then it's even more vulnerable.
Yeah, and so then B cells that see that site, the antibodies that recognize that site, they grab onto it, and then that B cell it grabs what it recognizes and it pulls it into the B cell, and that gives us a dose of vaccine.
Right.
And so the B cell that can grab that gets 20 times the dose of the strain-specific B cells that they don't get enough dose. And so your broad B cells, they're able to proliferate and thrive without being outcompeted by the strain-specific responses.
Yeah.
And so the results were pretty astonishing. In the pigs, the ferrets, we've done mice, rats, and even human immune organoids, which is they take lymph nodes from organ donors and they smush them up, and they are able to make these little like lymph nodes on a petri dish, and you can vaccinate them and see the responses of an adult, where you can get this ultra broad response. And what was attractive about the technology was, first off, you got to leave, you didn't have to do funky engineering on the proteins. You got to leave them as nature presented them. Because there were a lot of groups that being like, we just need to make the perfect spike. And I was like, there's no such thing. This is nature. It's an insane whack-a-mole problem.
It already exists, right?
Yeah, the site's already there. Yeah. It's just hidden in plain sight among all the components. So by mixing them together, we'll dilute out all the parts that are different. And reinforce the conserved site. And so we deliver them. And the other thing was it was very generalizable. So the same technology is working. Our flu program is a clinical candidate, so it's ready for human trials. We have the same technology we're developing for coronaviruses because they also have a conserved site to make a much more broad spectrum coronavirus vaccine.
So you're working on a broad spectrum coronavirus. You already have the flu.
Yeah.
And that's available somewhere in the world?
No, it's in manufacturing.
Oh,
so
that's
basically
the one that's almost ready for human trials.
That's right. And then we're applying the technology now to malaria. We're testing it on HIV. HIV is we're having problems displaying the proteins in a way that keep them stuck together, but it has these conserved sites. And then we're interested in there's a number of other pathogens that the whole problem is that they mutate quickly, and so the vaccines deteriorate. So we think we have a general tool where we could go after a number, not every pathogen. This is not this, but from the ones where they mutate to escape, we think we have a breakthrough that can be applicable.
So cool. So speaking for lay people, then basically, the universal flu vaccine is almost ready for testing.
Yeah.
Coronavirus, which is what people are the most afraid of, I think, given what's happened, how far along is that now?
I think it needs another year. So our portfolio, we'll be testing as early as December, but realistically in manufacturing, there's always delays. So I think we're probably looking at more like January, February, we start our human trial for universal flu. Coronavirus, during that same period, I'm estimating we have about 12 months of final engineering. We have completed studies on a Gen 1, and then we're doing these that safety optimization, for instance. So we prove that it produces broad responses. Now we're saying, okay, we have a whole bunch of diverse spikes, adding in a little mutation that makes it so they don't bind to the heart tissue, because we want to make it enhance the safety.
Right.
And so there'll be a cycle of testing there. And I think we have about 12 months later, we would start manufacturing, and the manufacturing process takes about 12 months. So it's about 2 years from now that one would go into human.
How do we enhance public trust? This is really the crux of this now, is that so many people feel like, well, I got the shot and I still got COVID.
Yeah.
And they, it's pretty high level. Everything we're talking about here is that they would understand, well, it mutated. Well, why should I bother? That kind of thing.
Yeah, so I think there's a couple different solutions based on a couple different reasons why people have confusion or vaccine hesitancy. There's a group of people that are going to go out and vaccinate, and they're going to take advice from ACIP and doctors, and that's an easy group. Those are essentially our allies in this. There's another group, which is on the other end of the spectrum, where I think no matter what you do, you're never going to talk them into vaccinating. And the truth is, it's a free country, and for herd immunity, for efforts of eradication, you don't actually have to have 100% of people vaccinated. And so there's a group where I'm like, I just don't think I'm going to reach them. And like, maybe that's okay that we have some diversity of.
What's the percentage you think for herd immunity if it's not 100?
It depends on the virus. So
okay. Let's say COVID.
I think COVID used to be 60. I think it needs to be higher because it's very infectious. Influenza, it's like 60%. I think you can establish herd immunity. Actually, during the pandemic, we eradicated one of the 4 lineages of influenza by accident. We used to have quadrivalent, 4-valent vaccines, because we had to target 2 different versions of HAB, the Yamagata lineage and the Victoria lineage. And during the pandemic, because of social distancing and masks and all these other measures, that stuff was designed to try to squish down transmission of coronavirus. But it also crazy squished down influenza. We had one of the the nicest, lowest incidence seasons.
I remember that. Yeah. And a lot of people said, well, we were all masking and we were staying away from each other. Do we really have to live like that forever?
I mean, people don't want to. And I don't think people should have to. I think we should celebrate the joy of life and being human. And I think we can build tools to do that. But one of the side effects of it is they actually rendered Yamagata lineage extinct by accident. So humans have wiped out smallpox. We've wiped out something called rinderpest. And we've also wiped out Yamagata by accident. So there's been 3 eradication successes that have happened in a remarkably short little slice of the long arc of history because of these interventions we have. Getting back to what we were talking about,
how to convince the public.
Yeah, to convince the public.
I hear what you're saying. There are always going to be people who are not convincible.
Yeah.
And there's nothing we can really do about that.
I think it's the big group that I think is convincible. And I think there's a couple of things that we should be doing. And some of that's some soul searching on the side of the industry. I think there's a lot of people who are like, I don't know, man, I get the flu shot or I get the coronavirus shot and I still got sick, right?
Yeah.
That's a group that, I think it's an easy group. If you come out with a vaccine that's not suddenly 30% effective, it's 95% effective. You're like, look, you take this. And so like a universal vaccine, which by the way, it sounds sexy to call it universal. The minute it comes out, it's just going to, or in the next few years, it will just be called the vaccine because that's the one that actually works.
Right.
And so people are more likely to take a vaccine if you can take it and it more reliably protects them, because it's confusing to take something and have it not work.
And it'll protect them from coronavirus and possibly some other things as well?
Well, the flu one is pan-influenza.
Okay, the flu one is
Coronavirus
separate, yeah.
pan-corona.
Yeah,
We are intending on creating combinations.
Right.
Because like MMR, V, and Tdap.
All this takes time. I really do see that, listening to the amount of work that goes into it. But I guess I would say if there were simple ways, this is not simple the way we've talked here, but if there were simple ways of communicating that to the public, almost like graphic stories for people to look at, graphic communication, like here's what we're creating. This is the part that never changes in that path. We can attack that. That's the weak part, et cetera. I think people would like to know because, and who knows how many people really want to know. But it seems like there might be some simple ways to communicate to people.
I like that.
Yeah, what it is you do.
I think so we have, for a publication that's coming out soon, we have a picture like that where we highlight the conserve site. I think public materials, I think...
it'd be great for doctors' offices and just online too, you know?
So I think that's part where it's really a much more effective vaccine that's also, you can take it all year round. You're not trapped having to take it within 2 months of vaccine season. It means that adoption's easier, it just works. You're like, I got that and I didn't get sick. If it works better,
people are going to want that.
That helps. Yeah. I think that you also have, the other reason I'm interested in patches is that I think there's a certain percentage of people who say they're vaccine hesitant, but they're actually just afraid of needles. And
there's a lot of, and they call it the jab. That's the slang for it. So, that doesn't make people feel very happy either. Like, God, I'm getting jabbed.
I think that being able to have a patch alternative for people, let's imagine you're a big tough guy from a vaccine hesitant state and you're actually deathly afraid of needles. What are you going to do? Tell people that? No, you're going to be like, no, I'm not going to take that thing.
I'm terrified.
Or it's a patch. You're like, oh yeah, it reminds me of nicotine patch. And so, so those are like technological things. Yeah. But I also think that we need to do a way better job of being transparent. Like during the pandemic, I had family members who, like, one of them works in a hospital. The other one's a psychologist. These are like sharp people. They're not anti-vaxxer, but they're concerned about their health and safety, and they're not convinced that they were getting good and reliable public information. And so they were contacting me, being like, I don't know, do you think this is safe? Is it safe for my kids?
I think I was contacting you at some point.
And I think like that's the big group of people that I think we need to be able to reach, and we need to be able, what my theory was, like, I'm just going to go say it like it is, and I'll give my assessment, but I'm not going to sugarcoat it. And I think that's what I would want from a field that I wasn't in, to know that someone's not like hiding things from me, to encourage.
The transparency is really important. People feeling like they're not being talked down to and that they're being told the truth.
And I got griped at because I went on public media and I said, look, I think that this is a vaccine that is like, the benefits far outweigh the risks for everyone except boys age 12 to 17. And I talked about that.
I remember when you said that.
And people complained about it, but I was like, I don't care. Like, if I had a boy of that age, I would not give him this vaccine. And like, who am I to not share that information with everyone else? And I think it had the effect of, first off, I think it put people of mind at ease because they don't think I'm trying to hide it. And I think that that's an important thing that this whole industry needs to do, to be like, look, here's how effective it is. Also, the effectiveness numbers were too high. I think when they said 94%, you're like, come on, like, people run these studies to time everything just right, and they take serum. And they just there's ways you can set these things up to make them perform a little better,
to make it sound better than it really is. Yeah.
And I don't think that's helpful.
No, I really think communication is going to be key, and I feel like, looking at everything you've done to get to this point, it's very exciting to think about where you're going next with it. And I just really appreciate you coming in and just downloading this huge amount of information, because, and who knows how many people are going to fast forward through the parts that are just like mind-boggling. But But that's the point. I think, people, whoever wants to learn this is going to learn this. And that's why I'm thinking, graphics would be nice for people to see. But thank you, Jake. It's really been great having you here. And we appreciate it a lot. I'm excited to see what you're going to be doing next. And I'll be watching the news to see when these are available to the public, too.
Well, thanks for having me.
And a patch, too.
Yeah.
I'm excited about patches.
Thanks so much for having me on. And I'm looking forward to being able to share those updates with you. Also, I like your idea, and you'll see me do something with, more efficient visuals so people can understand what we're doing.
Good, okay. All right, thanks.
Thank you.
Bye.